Hi, and welcome to the AP Biology Lab 6 Molecular Biology podcast. This is just kind of a short overview of the two parts of this lab. You can see right here we've got DNA, and that DNA, we're going to do two things with it. First of all, we're going to do what's called bacterial transformation, and what we're going to do in that is try to put the DNA actually inside a bacteria. And the other one that we're going to do is gel electrophoresis, and so we're going to take a section of DNA and then cut it into different fragment lengths and then separate those. And so let's go to the first one, bacterial transformation. In this picture right here, you can actually see E. coli bacteria that's growing on a plate. It's growing on some lauria broth. And so in each of these cases, you can see something different. And so this would be normal. This would be under ultraviolet light. These ones are producing a glowing protein, so they're glowing. That was actually extracted from jellyfish. And so that's going to be the bacterial transformation part. And then if we look over here, this is sections of DNA that have been separated according to their fragment length. And so let's start with the transformation. The one that we use in class is called pGlow, and this was created by, it was just artificially created by a company. And so let's look at the important parts of the plasmid. Remember, a plasmid is like an auxiliary section of DNA that does something else. And so in this case, they've engineered an origin of replication. That's where you're actually going to make copies of the DNA and make RNA from that. The other three genes on here that are important, this BLA, what it does is it actually it mediates ampicillin. In other words, it allows the bacteria to grow in the presence of an antibiotic. Over here, ARA-C, the function of that is to activate using arabinose, which is a sugar that's found. It actually makes a protein that then promotes the GFP. In other words, it starts this other gene, which actually makes the glowing protein. And so that's a lot of stuff. So let's look at what actually happens. Here we have 4 different plates, and so we're going to take regular bacteria, and in 2 of those plates, we're going to actually add the plasmid, and in 2 of the plates, we're not going to add it. And so we've got plus pGlow and then minus pGlow. In these, we're also going to have lauria broth and ampicillin. And so lauria broth, in this one right here, we should see bacteria growing. In this one, since there's ampicillin, we're not going to see bacteria grow unless they pick up that BLA. Which they won't if they don't have the plasmid. Next, we've got plus pGlow. Let's start over here, and that has lauria broth and ampicillin. In other words, these ones are going to be able to grow if they picked up the plasmid, but it's only in this one where we actually have nutrient. We also have that ampicillin, and the only ones that are going to be able to grow are those that have picked up the plasmid. And then finally, we have that arabinose sugar. And so this is the one where you could actually see that growth. Now, how do you get bacteria to be actually transformed? What you do is you heat shock them. So you'll actually put the bacteria in hot water for a period of time. You then add a transformation solution, then you add the plasmid, and then you just cross your fingers, because it usually doesn't work. But occasionally, some of the E. coli will pick up that plasmid, and we only need one of them to pick it up, because then we can grow them on a plate, we can have number colonies, and then we're going to have a bunch of glowing protein. So that's the bacterial transformation. There's not much analysis as far as that goes. It's just a hope that we can get the bacteria to pick it up. Let's look at the other one then. The other one is called gel electrophoresis. Gel electrophoresis, and this is a gel box. I've got one right here. So a gel box looks something like this. It's filled up with a buffer solution, and then your gel just kind of floats on that surface right there. The gel that we use is an agarose gel, and so the way to think about that is it's got macromolecules in it. It's got these bonds between them, and so the DNA, as it moves from one side to the other, it's going to separate, and it's going to separate according to its fragment length. And so how do you do that? First of all, you make a gel. You then get the DNA samples, and we'll actually add a restriction enzyme to them. And what a restriction enzyme does, remember, is it cuts the DNA in a specific part. And we use different restriction enzymes in each of those DNA samples that we have. The sample of DNA is always the same, but we use different restriction enzymes. One thing we use that is not pictured here is we actually use a DNA ladder. And what a DNA ladder is, it's a bunch of DNA cut into known base pair distances, and so we can actually compare that. We then just hook up some high voltage to it, and then we let it run. And what you get, let's look over here on this gel box, what you get is you'll load the DNA on this side. This is the tricky part, because you want to make sure you don't load it through the gel. But the DNA is going to migrate, since it has a negative charge, it's going to migrate from the negative side over to the positive side. And so depending on when you stop it, we're going to have those fragments at different lengths across the gel. So what does it look like when it's done? It looks like this. And so here's the wells where they were actually loaded, and here's the DNA fragments moving in the other direction. And so which of these fragments is the smallest? The one that moves the farthest distance. And so we can use a log scale to make something called a standard curve. And what we can do is measure the distances, and along that standard curve it's going to tell us how many base pairs it is long. So that's one way you could analyze the DNA. Another way that we'll use is a DNA ladder, and that's a known DNA that we run in another lane on our gel, and each of those fragments have a given length. In other words, one will be 100 base pairs, one will be 500 base pairs, and so then you can just compare across, and by comparing across we can see how long each of those fragments were. And so I think that's about it for molecular biology, and I hope that's helpful.